Protein translation. This presentation was made for an assignment for the module BMol 20090 at the University College Dublin in 2017 by Lily Whelan, Kimberlee Wiebe, Hareem Hassan, Shona Gorman, and Lily Cheung.
I will start with a brief historical synopsis of protein translation. Francis Crick's description of the central dogma of molecular biology originated the concept of mRNA in the late 1950s. This central dogma suggests that DNA turns to RNA to make a protein. Crick proposed the adopter hypothesis, which put forward the idea that there had to be a molecule that transported the amino acids from one location to the next, and put them in their correct sequence that was dictated by the nucleic sequence. Crick suggested that there were precisely 20 adapter molecules with this function, which Robert Holley later furthered and named the molecules transfer RNAs, or tRNAs for short. The process of translation was found through experimentation in a cell-free environment, which was made when the cell walls were broken down to release the cell's contents so that the cytoplasm that would remain in the cell would continue its capability to make a protein when RNA would be added. This cell-free environment allowed the researchers to study how RNA works and follow the movement of it without the distractions of other molecular activity. Nierenberg and Mathai chose E. coli to use to carry out this experiment. E. coli extract was added to 20 different test tubes, with every test tube containing a mixture of the 20 amino acids, but one amino acid, a different one in each test tube, was radioactively tagged, so that the reaction could be monitored and followed. After this experiment had cracked the first word of the genetic code, at 3 a.m. on May 27, 1961, scientists around the world began to translate the unique code for each amino acid, aspiring to read the entire genetic code of all living organisms. These 20 scientists called themselves the RNA Time Club, 8 of whom won Nobel Prizes for their contributions in discovering and determining RNA and its functions. They determined the idea of the codon, 3 nucleotides combining 4 codons and 3-letter codes yielded 64 possible combinations, sufficient to describe 20 amino acids. They went on to discover the codes for other amino acids, for example, AAA for lysine and CCC for proline. Replacing one unit of a triplet code with another nucleotide yielded a different amino acid, for example, GW produced valine. By 1966, Nierenberg had deciphered the 64 RNA 3-letter code words for all amino acids. So translation is a step in protein synthesis. When the cell finds itself at translation, mRNA has just been previously transcribed. This means that the genetic information necessary for the production of protein has been taken down and recorded in the previous step called transcription. Now during translation, it has to translate what has been transcribed. The mRNA molecule, which stands for messenger RNA, is holding this information. This information is held in the form of nucleotides. Nucleotides are made up of one 5-carbon sugar, a phosphate group, and a base, as shown in the diagram. Every group of 3 nucleotides codes for one amino acid. These 3 nucleotides are called codons. So basically, 3 nucleotides is equal to 1 codon, which is equal to 1 amino acid. This all occurs in the ribosome of a cell, and ribosomes are located in the cytoplasm of prokaryotic and eukaryotic cells. They can be also found in the endoplasmic reticulum, which is often shortened to the ER. There are 2 types of ER, a smooth one and a rough one, and these ribosomes are located in the rough ER. So a ribosome has 2 units, a small one and a large one. It is globular in shape, as you can see from the diagram. Here you can see mRNA acting like a chain passing through the ribosome. Ribosomes are different sizes in prokaryotic and eukaryotic cells. Even though we are only focusing on translation in eukaryotic cells in this presentation, I will discuss the different sizes in both cells. So as I said before, there are 2 subunits in the ribosome. For prokaryotes, one is 30S and the larger 50S. This actually means that the whole of the ribosome is 70S. In eukaryotes, the smaller subunit is 40S and the larger 60S. This equals to 80S. Of course, we all know that 30 plus 50 does not equal 70, nor does 60 plus 40 equal 80, but S is not a metric unit. S is actually a rate of sedimentation. It is a measurement used back in the day of how fast cellular structure is sediment in centrifugation. The Spedberg is actually a measure of time. It is defined as exactly 10 to the minus 13 seconds.
What is activation? Activation is the establishment of the genetic code through a process where enzymes called aminoacyl tRNA synthetase connects the carboxyl group of amino acids to the corresponding nucleotides at the 3' OH end of the tRNA by a covalent bond. When tRNA is charged and has an activated amino acid linked to it, it is ready for initiation. There are 20 amino acids, each recognized by a specific aminoacyl tRNA synthetase. The specific aminoacyl tRNA synthetase, also called activating enzymes, catalyze the reaction. These enzymes are all different, but they are used to catalyze the synthesis of aminoacyl tRNA using ATP. For example, when the mRNA 5' to 3' strand is CUC, it pairs with the anticodon base pair GHE on the tRNA. This tRNA would have a leucine amino acid attached to it because the mRNA is CUC, which codes for leucine as seen here. During the reaction, the amino acid with a specific R group is attached to a tRNA that corresponds to the specific R group that the amino acid has. The amino acyl tRNA synthetase is the catalyst in this reaction. ATP will be converted to AMP and pyrophosphate. The amino acid is covalently bonded to the tRNA, giving a product of the amino acyl tRNA. The pyrophosphate is hydrolyzed, which gives the effect of 2-ATP being used in this reaction to form amino acyl tRNA. The sum of these activation and transfer steps is amino acid plus ATP plus tRNA producing amino acyl tRNA plus AMP plus pyrophosphate. This demonstrates the first ATP is consumed in forming the ester linkage of amino acyl tRNA, and the second is essentially consumed in driving the reaction forward using AMP and pyrophosphate. Now the amino acyl tRNA is ready for initiation.
The initiation of the protein translation is when the ribosomal tRNA complex is complementary attached with the initiation codon on the messenger RNA. For the initiation process, there are a few key characters. These characters include a ribosome, a strand of messenger RNA, a transfer tRNA molecule which is holding the sequence for the first amino acid in the protein sequence, which is mostly the methionine. These parts come together to form the initiation complex, which is the initiation molecular machinery for protein translation. There are 6 steps in initiation. The first step is the breakdown of ribosome into its subunits, formation of the ternary complex of tRNA, formation of the 43S pre-initiation complex. 43S complex is brought to the 5' cap, the scanning, and the assembly of the ribosome. Types of initiation in eukaryotes. There are 2 types of initiation. In the cap-dependent initiation, the 43S complex carrying the small subunit of ribosome and the initiator tRNA is bound to the 5' end of the mRNA molecule. This is called the 5' cap structure. This structure is present on the 5' ends of messenger RNA. Guanine nucleotide is bound by 5' to 5' triphosphate linkage by methyltransferase on the 7th position. In the cap-independent initiation, there is no need for the ribosome to start scanning from the 5' end of the mRNA. Rather, the 43S complex is brought to the start site by certain factors called the IRES transacting factors. Translation initiation setup has become very complex with evolution and now contains up to 11 factors. The initiation factors are the factors that are binding to the small subunit of the ribosome during the initiation. Their function is to ensure that the tRNA is brought to the P site of the ribosome to the initiator codon on the mRNA. We'll now look at an animation that will illustrate all the sub-steps of initiation. There are multiple and distinct steps. In the first step, the ribosome is broken down into its 40S and 60S subunits. The initiator factor EIF3 binds on the small ribosome subunit in order to prevent the 2 subunits from rejoining. In the second step, the methionine carrying tRNA molecule is delivered to the P site of the 40S ribosomal unit by the interaction of EIF2 and EIF4 initiating factors. The 43S complex is brought to the mRNA by EIF4 initiating factors to the 5' cap. The complex travels along the mRNA until the methionine tRNA base pairs with the initiating codon. This is known as the scanning model. The EIF-IA factors help the complex to bind at the initiating complex. After base pairing, the large subunit is attached at the site, forming an active ribosome. The factor EIF5 hydrolyzes the GTP into GDP, and factors bound to the small subunits are released. The ribosome is now active and ready for elongation.
Elongation. Elongation is the third stage in the protein translation process. In this stage, amino acids are transported to the ribosome by tRNAs and are then linked together, forming a chain. Elongation is basically the lengthening of the amino acid chain. In biology, elongation refers to a phase or stage in the protein translation process in which a genetic code carried by the mRNA is read and decoded to produce a sequence of amino acids in a polypeptide chain. There are 3 different sites associated with the large ribosomal subunit. The A site, otherwise known as the amino acyl site, is the entry site for new tRNA. The P site, or peptidyl site, is occupied by peptide tRNA. This is the tRNA that carries the growing peptide chain. The E site is the exit site for the tRNA after it is finished delivering the amino acid. What happens in elongation? mRNA and tRNA are the main molecules needed in this stage, as well as the ribosome itself. mRNA acts as a substrate for translation and contains codons which encode for specific amino acids. tRNA, otherwise known as transfer RNA, contains an anticodon which is complementary to the mRNA codon to which it binds. Attached to the tRNA molecules is the corresponding amino acid. For example, the anticodon CGA has alanine as the corresponding amino acid. The steps of elongation are as follows. Firstly, a tRNA carrying methionine binds to the P site of the ribosome. Beside it, a new codon is exposed in the A site. A second tRNA amino acid lands in the A site of the ribosome. The anticodon attached to this second tRNA will be complementary to the exposed codon. As seen in this animation, if the second codon is CGU, then the second anticodon will be CGA. The specific tRNA will be carrying the amino acid alanine. An enzyme in the large subunit of the ribosome transfers methionine from the initiator tRNA to the alanine bound to the second tRNA, and a peptide bond is formed. The ribosome then slides onto the next codon on the mRNA. This process shifts the initiator tRNA to the E site and the second tRNA into the P site. Once again, a new codon is exposed in the A site. After giving its amino acid to the newly formed peptide bond, the initiator tRNA will exit the ribosome via the E site. This process will continue until a stop codon is reached.
Termination is the process that terminates protein synthesis in translation. So why does termination occur? Well, it takes place for several reasons. Firstly, to stop protein synthesis, which took place previously during initiation and elongation. Secondly, it is necessary so that the polypeptide chain which has been formed is released. If it did not occur, the polypeptide would not be liberated from the translation assembly. Lastly, termination is key to the disassembly of the ribosome complex. There are two main steps involved in termination. The first step is the termination reaction, which is the central step as its main function is to release the polypeptide chain. The second step is the post-termination reaction, and it involves the release of the ribosomal complex, the tRNA and the mRNA. The first step, which is the termination reaction, is initiated when one of the stop codons, either UAA, UAG, or UGA, enters the A site of the ribosome. None of the stop codons have a corresponding amino acyl tRNA. Here we can see the stop codon entering the A site. There is no amino acyl tRNA to recognize the stop codons. This means that they can't be recognized and that protein synthesis has stopped. In place of an amino acyl tRNA, protein factors called release factors recognize the stop codons and bind to them in the A site. In eukaryotes, there are two release factors, ERF1 and ERF3. They each play a different role in the termination reaction. ERF1's function is to recognize all three of the stop codons. ERF3 triggers the activity of ERF1 and it aids ERF1 to release the polypeptide chain. ERF1 has some similarities to tRNA because it positions itself in the same location on the A site that the tRNA would. ERF1 also activates the hydrolysis of the ester bond that is linking the polypeptide chain to the peptidyl tRNA. ERF3 is a GTPase. A GTPase is an enzyme that can bind to and hydrolyze GTP. GTP is not unlike ATP. Once the ester bond has been hydrolyzed, the polypeptide chain is liberated and leaves through the exit site on the ribosome. When the termination reaction is completed and has released the polypeptide chain, the second step, the post-termination reaction, can begin. This is when the translation assembly comes apart. The components which must dissociate are the large and small subunit, the mRNA and the deacylated tRNA. The polypeptide chain will undergo some modifications once it has been released from the ribosome, for example, folding. ABCE1 is an ATPase and it binds with the ERF1. ATP is hydrolyzed. This enables the 60S and 40S subunit, the tRNA and the mRNA to all dissociate. The large and small subunit are recycled.